Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to understand what are extrinsic factors. In the previous lecture we have already covered the intrinsic factors. So in this particular lecture we will have a detail of what is extrinsic factors. So we all know that the microorganism means the ability of microorganism to grow, or you can say to multiply in food, is determined by the food environment as well as the environment where the food is stored. Okay, so on the basis of two factors it's depend, means how the microorganism will grow. It totally depends on two factors. The first one is the food environment as well as the environment where the food is stored, means the storage area. That is the reason in my lectures I always say that storage play a very important role whenever a microorganism growth is concerned, because if the area is clean, if the area is sanitized, if the storage area is free from any sort of microbial contamination, then yes, your food is safe. But if the area is not clean, it's unhygienic, the area storage area is having a lots of you can say dirt, so eventually there is a great chances, or of course there is more and more of microorganism. So it is very important for a industries, for a food manufacturing industries, to have a area that is free from any sort of microbial contamination, means the area should be clean, the area should follow the GMP's guidelines, the area should follows the GLP guidelines and many other regulatories guideline. So it is very important to have an area that is clean and properly sanitized. So we have already covered the intrinsic factors. So again, I'm just repeating. So intrinsic factors, these are the factors that is related to the food itself. For example, nutrients, water activity, pH, redox potential, etc. But as far as the extrinsic factors are concerned, so these factors which are controlled through the external condition, they are referred to as extrinsic factors, or you can say the factors in the environment and the external to the food which affect both the organism and the food shelf itself during the processing and storage is known as the extrinsic factor. Now, the extrinsic factors includes the storage temperature, storage relative humidity, atmospheric gases, microbial activities. So, we will going to understand each one in detail. Now, temperature. So, students, we all are aware that temperature play a very important role whenever the growth of microorganism is concerned. Okay, so the storage temperature simply means the foods that are exposed to different temperature from the time of production until the time of the consumption. Okay, so microbial growth is accomplished through a enzymatic reaction which is depend on the temperature. And this microbial group, for example, saccharophiles, mesophiles and thermophiles, it is being divided into three groups. So at every 10-degree rise, it doubles the catalytic rate of enzyme, and at every 10-degree decrease it reduces it to the half. Okay, let us discuss this in detail. So first of all, the enzymatic reaction, or you can say the micro and the microbial growth, they are largely affected by the environmental temperature. That is the reason I am saying that whenever any food it is exposed to a different temperature from time of production to the time of consumption, so it plays a very important role, means at what temperature the food is actually produced and at what temperature or at what time the food is actually consumed. So these two factors play a very important role. Okay, so the growth of temperature for yeast and mold demonstrate a broad range of 10 to 35 degrees centigrade. So based on the temperature, means if we see this, that according to the temperature, the bacteria, if we take an example of bacteria, so it is divided into three categories. The first is psychrotrophs, or you can say the first one is your psychrophiles, second is mesophiles, and third one is your thermophiles. So psychrophiles are those bacteria that are cold tolerant. Cold tolerant simply means that can survive at a very extreme cold conditions, and they can grow at the temperature range of 0 to 20 degrees centigrade, and this includes pseudomonas species and enterococcus species. So in MCQs, in interviews, this questions generally asked that name any two psychrophiles bacterias, or what do you understand by psychrophiles. Or the third question can be made here is at what temperature these bacterias can grow. So that range is 0 to 20 degree centigrade. Now comes the mesophiles. So mesophiles, they are microorganism, bacteria that can grow between 25 to 40 degrees centigrade. So at what temperature these mesophiles can grow? So they can grow at a temperature between 25 to 40 degrees centigrade, with the optimum growth of temperature that is close to 37 degrees centigrade. And these microorganisms, they include the species of Salmonella, Staphylococcus, Clostridium, Shangela, and Bacillus. So these are the species. The same question can be asked from this as well. What are mesophiles, or at what temperature, or what is the optimum temperature for mesophiles to grow? Give any two examples or three examples of mesophiles, then this is the answer. Now the third one is the thermophiles. So thermophiles, these are the organism, or you can say these are the microorganism that grow at a very high temperature. Means whenever I'm saying very high, means very extreme high temperature, that is above 45 degrees centigrade, with an optimum growth temperature between 50 to 70 degrees centigrade, and this includes Bacillus, Clostridium, Geobacillus. So uh, so these are the three groups of microorganism depending upon the temperature. Okay, now moving further, that is the effect of temperature on the bacterial growth. So according to this graph, we can see that one side it is the growth rate, and second is the temperature. So as a very initial, means whenever there is an initial, that is minus 10 to 10 degrees centigrade, which organism, microorganism is showing a proper growth, that is a psychrophiles. So at this temperature, psychrophiles are the organism that grow. Now your next is psychotrophs. So psychotrophs, example E. coli, so they can grow at a room temperature. So psychrophiles grow at the freezing point, whereas the psychotrophs, it grow on room temperature, that it require 10 to 35 degrees centigrade, and at this temperature they show their optimum growth. Then at from 40 degrees centigrade to 55 or 60 degrees centigrade, mesophiles grow. Okay, then thermophiles above that temperature, means the optimum range can go at a very high temperature, like up to 80 degrees centigrade. Then at this rate, or at this temperature, thermophiles show their major growth. Then comes your extreme hyperthermophile at like boiling temperature. So at this temperature, hyperthermophile bacteria can survive. So the simple thing is that at freezing point, Psychrophiles bacteria are there which can survive even at the freezing temperature. That is the reason whenever you store anything in refrigerator for a longer and longer period of time, let's say 2 years, then of course it will get deteriorate, because at that temperature also microorganism growth. If you put any food at a room temperature, then after few hours the microorganism attack that food, particular food items, and they can deteriorate the quality of the food, means the food gets spoiled. So why I am saying this, because at every temperature there are certain microorganism, or there are certain bacteria that can easily grow. Okay, so that is the reason effect of temperature on a bacterial growth is shown by this particular graph. So this graph is very important. If you want to take a screenshot, you can simply take a screenshot of this. That is very helpful, that will be very helpful for you. Now let us take a note and to understand this, that the effect of temperature on microbial growth also depends on the environmental condition as well. Means now the effect of the microbial growth, so at which how it depends, so it depends upon the environmental condition also, like the growth factors, in the medium, even the pH of the food, water activity. So all these things I have discussed in detail in my previous lecture, so you can go to that lecture and get and have an understanding of what is the growth factors in the nutrient medium, what is the pH of the food, and how it is the microbial growth is depending upon pH of the food, and same as well for the water activity. Now coming to the relative humidity, or you can say the storage relative humidity. So storage relative humidity is the measure of water activity of the gas phase. Okay, so the simple answer is means if in examination or in interviews, if the question generally comes, what is relative humidity? So the relative humidity is the measure of water activity of a gas phase, or you can say the relative humidity is the amount of moisture in the atmosphere of food environment. So the relative humidity, it can influence the water activity and water activity level on the food, and hence can influence the growth of microorganism. Means if for example dry grain stored in an environment with high relative humidity will take up water and undergoes mold spoilage. Apart from this, if the relative humidity is very high, means high relative humidity, it influences the water content of the food. Means whenever, I'm just writing, whenever the relative humidity is very high. Okay, so let us see this. Whenever the relate sorry whenever the relative humidity is very high, okay, so what it does, it influencing the water content of the food. So what it is doing, so it is influencing the water content of the food when the relative humidity is very high. Okay, and at high water content it promotes the microbial growth. Okay, so whenever the water content is high, so of course water is the hub for the microbial growth, so it is influencing the growth of, or it is promoting the growth of microbes. Okay, so when the food is again stored at a very high relative humidity, so what changes it does, so it has a dormant spores of bacteria or fungi to germinate, and once they are actively growing, once these spores of bacteria and fungi, they are actively growing, so they produce water as an end product of their for their respiration, and hence after this they increase the water activities of their own. means the bacteria will increase the water activity of their own. This favors the growth of high water activity requiring for the bacteria, and yes, in that particular case the increase in the spoilage of the food occurs. Again I am repeating that whenever the food is stored at a very high relative humidity, matlab relative humidity is very high, so in that particular case, the dormant spores of bacteria, or you can say fungi, they get germinate or they can grow. Once they are actively growing, okay, so they produce water. After they are producing water as an end product or for their own respiration, so they will increase the water activity of their own. Yes, and after increasing the water activities of their own, they are influencing the growth of you can say sorry this favors the growth of high water activity that is requiring for the bacteria. And yes, at that particular point, when the bacterial activity is at a very growing at a very high rate, then of course there is an increase in the food spoilage. Therefore, a dry condition are considered better for food storage than as the moist condition. So here one question generally arises, so the answer I have already told you, but I am just writing the question. Why why dry condition, okay, why dry conditions are considered better than moist condition? So the answer is very same that we have just uh, I have just explained, sorry. So this is the relative humidity. Now the presence and the concentration of gases in the environment. So see, presence or absence of gases, it affects the microbial growth, and of course it also affects the type of microbial population. For example, carbon dioxide, it regulates the cell growth of certain bacteria. Okay, what CO2 is doing, so they are regulating the cell growth of certain bacteria. Then if there is a partial pressure of CO2 gas, or so they increase if the partial pressure of CO2 that is carbon dioxide, it gets increased over a very critical level, so yes, the metabolic activity will be retarded. And if there is a delay effect of CO2 increases with the increase in the concentration. So the carbon dioxide is used in packaging of food items in order to control the growth of microbes. Then how it is done? So see, gases like CO2 or ozone and oxygen, they have a direct toxic effect that can inhibit the growth and proliferation. That is the only reason CO2's gas is used in packaging, that is atmospheric packaging, and when the food is being packed. So why? Because they have a very direct, or you can say they have a very toxic effect on the growth of a microorganism, and they can inhibit the growth and proliferation of different kinds of microorganism. Ozone and oxygen, again, they are very highly toxic to anaerobic bacteria, and carbon dioxide is affected against the obligate anaerobes. Now, foods affected by various groups. So there are certain groups through which the food are various, means they are affected in a various group, like anaerobic, or you can say the facultatively anaerobic. There is your micro aer- aerophilic bacteria, then it is a aerobic bacteria, then it is again the aerobic mold. So the first one is anaerobic. So these are the spore formers, and they are likely to grow in a canned food. So whenever the food is canned, means in nowadays there are different canned food that are available in the markets. So in that particular food items, how they can deteriorate, or how they can be they can become unfit for human consumption if anaerobes big bacteria they can grow. So in this way, the anaerobic are the microorganism that is capable of forming the spore. That is the reason they are also known as spore formers, and they are likely grow in the canned food. Then is your aerophilic bacteria. So these are the bacterias, they are mostly grow in the vacuumed packed. So since there are again food items that are vacuumed packed, in that condition also the bacteria can grow. Which type of bacteria? The microaerophilic bacteria. And how? Since they have a low oxygen tension, so that is the reason they can easily grow. Okay, then is your aerobic bacteria. So aerobic bacteria, they are likely to grow on the surface of raw meat. Means whenever the meat is not cooked, after slaughtering the raw meat, on that particular meat, or on that particular surface of the meat, aerobic bacteria grow. Then is your aerobic molds. So they generally grow on a dried or a salted product. Okay. So in that particular condition, these type of bacteria can grow. Now it is very important to understand the regulatories, industrial and international implications. Okay, so many of the intrinsic factor, especially whenever we are doing any heat treatment, or whenever the food is stored, so the food storage temperature requirement do all fall under the federal, state and international regulation. Okay, the first one is heat treatment. So, commercially sterilization of thermotically sealed food product domestically if manufactured or imported into the US is regulated by FDA, that is Food and Drug Administration, and these regulation covers the very low acid canned food, that is known as LACF, and sometimes they can ask you to expand LACF. So what is LACF? So LACF is the, I'm just marking it down for you. LACF is the low canned food, and generally they have a pH of sorry they have a pH range between 4.6. Okay, so they have generally a pH of 4.6, and they are considered as an acidified food. They have pH range of 24.6. So now the second one is according to the again I would like to explain what is pasteurization. So whenever the milk is raw, means whenever the meat is raw, so there are great chances of microbial contamination or the growth of microorganism. That is the reason milk is pasteurized at a very high temperature for a less period of time. The pasteurization and is done, and in that particular case, why pasteurization is done, just to, you can say, inhibit the growth of microorganism. So at that particular point of time, pasteurization is done, and this inhibits the growth of different kinds of microorganism that is present in the raw milk. So without pasteurization, milk is not considered to be consumed. Why? Because there are high chances of microorganisms. Whenever I'm saying high chances of microorganisms, then of course pasteurization helps to heat the meat, at a very specific temperature, that according to a set of time, and that is for 15 minutes. And at that particular temperature, the milk is pasteurized, and this what it does, so it generally kills or inhibits the growth of microorganism, making the milk free from any sort of microbial contamination. And yes, the milk can easily be, you can say, used by the people. So in different books there are different temperature written, and the milk is actually boiled at a temperature of 72 degrees centigrade for 15 seconds. Sorry, I've just mentioned 15 minutes, so sorry for that. It's 15 seconds. Okay, so at this particular temperature, the all the pathogenic microorganism in certain beverage and food, sorry, milk product, it get deteriorated, it get destroyed. And this pressurized temperature, sorry, the pasteurization, it is named after a French scientist we all are aware of, Louis Pasteur. So he first demonstrated the fermentation of wine and beer, and he was thinking how to prevent this beer and wine from microorganisms. So he used it technique by heating the beverages for about 135 degrees Fahrenheit for few minutes, and after that the pasteurization of beverages and milk get into uh, is being done. So according to the milk pasteurization regulation, that defines the grade A pasteurized milk accordance, it is necessary to ensure that every particle of milk is heated, okay, to appropriate temperature for an appropriate time, that I've already told you. Furthermore, that the equipment used meets the strict regulatory testing and control to avoid the any risk of cross contamination with raw product or risk of post pasteurization contamination. Let's take an example. You purchase a milk from a market, okay, and if you store that milk product or a milk, sorry, in an equipment that is not clean, and you are doing heating, then what will happen? Of course your milk will deteriorate. Why? Because of the cross contamination. So it is very important for a person who is doing pasteurization that the equipment should also be clean, should also be properly sanitized, so that there should be no risk of cross contamination. Okay, if you want to avoid any risk of cross contamination, so it is very important, it is very important that the equipment should be clean, so that there should be no risk of cross contamination, and in this particular way the milk get free from any sort of microbial contamination. Now temperature required for the food storage, let us quickly complete this. Maximum regulatory storage temperature it requires, requirement have been traditionally set at 7 degrees centigrade, that is 45 Fahrenheit, by the state and federal regulation. For a number of commercial food products that includes the milk and meat and sea products. So if you see at which what is the storage temperature that is required for the storage of different kinds of food product, that is milk, meat and seafood, so it is 7 degrees centigrade, that is being set by the state as well as from by the federal regulation. Okay, now because of the concern about psychrotrophs peak growth that grow at a very initial temperature, means at a very low temperature, you can refer to this graph for better understanding. Psychrophiles, so they grow at minus degree centigrade, and for the, so coming back to that, because of this concern, and that is a psychotropic growth, so certain pathogens of certain pathogen, so the FDA recommended a temperature for food storage in retail established that is reduced from 7 to 5 degrees centigrade. So if in examination or in interviews, if they ask you what is the storage temperature, that what is a recent storage temperature at which the food is stored, so it is 5 degrees centigrade. It is not 7 degrees centigrade, but yes, it was. But after the concern about the cyclotropic growth, so this change 7 changes to 5 degree centigrade or 41 degree Fahrenheit or below to that. So that brings to the end of the lecture. I hope you have understood the lecture. Again, if you have any queries or any questions, so you can simply leave your queries in the comment section below. In the next lecture, I'll be answering the queries. For the intrinsic factor, you can refer to the previous lecture for better understanding of the whole topic, that is intrinsic and as well as extrinsic factors that affects the growth of microorganisms. So thank you, and don't forget to follow ITLS Academy on different platform. You can also visit our website, that is www.itlsacademy.com. This is our helpline number, that is 7080833450. You can simply call on this number and get updated. And thank you for watching. Please do like, share and subscribe. And at last, don't forget to press the bell icon so that you can get a latest update on the lectures. And again, if you want to enroll in our courses, so you can directly reach out to us through our website or through this helpline number. Thank you once again.